Hello, everyone. Thank you so much for having me. I've loved SerpentineDays webinars for a variety of years, so it's nice to be able to share my work. I'll be talking today about our work in the Oman Active Alteration Microbial Observatory, where we're measuring dissolved inorganic carbon and other carbon sources with 13C stable isotope probing. And our main finding and the title of the preprint that came out of this work is Dissolved Inorganic Carbon Supports Robust Anabolism and Methanogenesis in Actively Serpentinizing Rocks. And so just to quickly summarize, we're studying serpentinization as the reaction of hydrogen or of ultramafic rocks with water, which produces hydrogen gas both through the serpentinization reaction and the formation of secondary minerals such as brucite. It increases the pH of the subsurface groundwater. And we care about this as microbiologists because this process fuels persistent microbial ecosystems in the absence of sunlight. And so much of the early work in this system in Oman was funded by the NASA Astrobiology Institute, the Rock-Powered Life astrobiology node, because evidence of serpentinization is widespread in the solar system. And so NASA was interested in this process as a prime target for life detection missions. And they've sent a variety of probes and rovers to look for serpentinization processes throughout the solar system. But now much of our work is focused on these two processes of carbon sequestration and geological H2 extraction. For the carbon sequestration approach, it's referred to as mineral carbonation, where CO2 is captured from the atmosphere, injected into the hyperalkaline fluids in the subsurface, where it precipitates as calcium carbonate. And for this process, we are interested in the microbial processes, which I've extracted here as these circular arrows that might turn dissolved inorganic carbon into methane, which would potentially inhibit our ability to sequester carbon in the subsurface. This is a bit difficult to study because, as we know, serpentinizing systems are rich in methane on their own. They're producing methane abiotically, and so the methane effluxes that you can measure at the surface are both integrated from the biotic forces and the abiotic forces. And so we get to this question of how do we measure the biological contributions to these, what we're calling invisible metabolites, where we're not entirely sure what proportion of them are produced by microorganisms and what proportion of them are produced by the serpentinization process in general. This is also true of geological hydrogen extraction, where hydrogen coming from the subsurface can be captured and turned into energy, but microorganisms can intercept some of this hydrogen and turn it into water by oxidizing it. And so similarly, this is an invisible process because we can't measure the water that is produced from hydrogen oxidation. And so there's an open question of how much of what makes it to the surface is representative of the true production rates in the subsurface. And so we study serpentinites in an analog system in the Samail ophiolite of Oman, which is the largest serpentinizing terrestrial body on Earth. And here you can see it's a very dry climate, and where this road cut has been drilled, you can see that just beneath the oxidized surface, there is this beautiful dark green and black peridotite subsurface. And the fact that it's a dry climate means that there are minimal terrestrial inputs of organic carbon into the system, which makes it ideal for studying serpentinizing carbon cycling in isolation from photosynthetic inputs. And so in 2017, the Oman Drilling Project and ICDP established this multi-borehole observatory, and I'll be talking about these three wells today. They're called BA1B, BA3A, and BA4A. The IDs are not as important because I'll be describing them shortly, but these are all in close proximity to each other. And in 2023, we accessed these boreholes with a subsurface discrete interval water sampler. And so we noticed a gradient of serpentinization across these wells. Starting with BA1B, we saw mildly alkaline fluids where the pH is still alkaline, but closer to neutral between 7 and 9. And the amount of cells that we counted across these different depth intervals were on the order of 10 to the 6th to 10 to the 5. In the BA4A, moderately alkaline fluids, the pH is between 9 and 10 with similar cell densities. And in the hyperalkaline fluids of BA3A, we see a much higher pH between 10 and 12. And at these hyperalkaline conditions, we start to see cell concentrations drop dramatically to 10 to the 4. And so we think this is related to the extent of mixing with meteoric waters, where the greater the mixing extent, the lower the pH, and the greater the oxidant availability, the more these groundwaters can support microbial ecosystems. But what kinds of microbial ecosystems can these groundwaters support? We did some preliminary 16S sequencing and found that methanogenic archaea are very abundant members of the community. They range between 20 and 50% of the total population of the microbial community, shown here in this light blue. This is a genus Methanobacterium, which has been studied in depth in this ecosystem before, and there's been a lot of work done on the genetics of Methanobacterium. But it's interesting to see that it can comprise as much as 50% of the microbial community in unaltered waters. And so the questions that this work addresses is, how does microbial activity map onto the geochemical gradient we see? What are the rates of microbial activity that these waters can actually support? And more importantly, what are the carbon sources that support life in the alkaline and hyperalkaline subsurface, where organisms are limited for both organic carbon, because this is a organic carbon limited environment in the dry conditions, and also limited for inorganic carbon because of the hyperalkaline conditions. So to get at this question, we use 13C stable isotope probing to reveal the rates at which carbon is transformed. And so what this process involves is taking a 13C labeled substrate, monitoring its assimilation into cells, and either through catabolic pathways, such as the production of 13C labeled methane, or through anabolic pathways, such as the production of 13C labeled biomass. And I'll start by talking about the anabolic pathway and anabolic rates. And so we measure rates of microbial biomass synthesis using nanoscale isotopic imaging of 13C incorporation. What this means is we take groundwater from our 3 different depths that we sampled, shallow, medium, and deep, from our 3 different wells, BA1B, BA4A, and BA3A, which again are going along that gradient of alkalinity. And we incubate these vials with one of 3 different 13C labeled carbon sources, 13C bicarbonate, 13C acetate, and 13C formate. And we chose 13C acetate and 13C formate because acetate and formate are other methanogenic, potential methanogenic substrates that could be involved in methanogenesis in the absence of abundant dissolved inorganic carbon. So there's this hypothesis that if organisms are limited for DIC, they might supplement their metabolisms with small molecular weight organic acids such as acetate and formate. So we take these cells, we incubate them for a certain period of time, we spin them down into an epitube, we spot them onto a specific type of aluminum slide, and then we put them on a nanoscale secondary ion mass spectrometer, or NanoSIMS. And what this involves is a primary ion beam of cesium that ablates the sample away and releases secondary ions 12C and 13C. And the data product that we are able to get from this is very interesting. It's essentially a single cell resolution isotope image of the sample, where the brightness of this image corresponds to the abundance of the heavy isotope that we labeled over the light isotope. And so we're able to get single cell measurements of anabolic rate. And so this also yields carbon source specific rates of carbon assimilation and biomass synthesis, because we're able to relate these single cell isotope ratios to the carbon source that they were, the carbon label that they were supplied with. So what this data looks like, I'm going to present this plot slowly because there's a lot going on, but across the x-axis, we're looking at cell-specific carbon assimilation rate in femtomoles of carbon assimilated into a cell per day. So that's just the amount of carbon from the 13C source that the cell is assimilating into its biomass. And we're looking across our mildly alkaline, moderately alkaline, and hyperalkaline wells at 3 different depths at 3 different carbon sources: bicarbonate, acetate, and formate. So the first bit of data I'll show is from the mildly alkaline wells. Here we're looking at distributions of single cell carbon assimilation, which is what these distributions show. And each of these little dots underneath the distributions corresponds to an individual cell that we measured. Whether the dot is black or red is referring to whether the isotope label was sufficiently detected. And so you can consider the red points here as inactive cells or cells that weren't active on a time scale that we could measure. The other thing you can notice across this BA1B condition is the difference of carbon preferences, where bicarbonate is preferentially assimilated because it's further to the right, then followed by acetate, then followed by formate. You might also notice that there doesn't really appear to be a depth effect here, where the depth of sampling between shallow, intermediate, and deep doesn't really affect the anabolic rates we're measuring. Next, we're looking at the moderately alkaline wells, and here we see a similar pattern where bicarbonate is preferentially assimilated compared to acetate, which is preferentially assimilated compared to formate, with formate having the greatest abundance of inactive cells. And finally, with the hyperalkaline fluids, we also see a similar pattern with even at pH 10.5, bicarbonate is preferentially assimilated compared to acetate, compared to formate. But here we are starting to see a bit of a depth effect. So across the shallow, intermediate, and deep conditions, we're seeing this gradient of microbial activity where organisms in the deeper samples are much less abundant, but also much less active. And indeed, in some of these incubations, such as the acetate and formate incubations of the hyperalkaline fluids, we're unable to detect any microbial activity whatsoever. And so here we're really nicely demonstrating this gradient of microbial activity across both depth conditions, demonstrating the preferred carbon sources of these organisms, which appears to be bicarbonate, and showing these rates at the single cell level and showing these distributions of microbial activity across an entire population. And so with that in mind, we then were curious about the catabolic rates of these organisms. So we know how quickly they're producing new biomass, but how quickly are they actually turning that 13C into methane? We took these incubations and measured the methane isotopes with a Caro gas isotope analyzer. And our results supported our anabolic rate measurements in that we found that the vast majority of biological methane comes from dissolved inorganic carbon. And so here we're looking at the fraction of methane that is sourced from the 13C probe as a relationship between 0 and 100%. And you can see that in the bicarbonate conditions, where we're able to detect methane, the fraction of that methane that came from bicarbonate is well over 80% in most conditions. Of course, it is the lowest in the hyperalkaline conditions, up just above 75%, which is still significant, but it does indicate some contributions of formate and acetate to the production of methane, which we're actually able to detect only in the shallow fluids. Where the data has an asterisk indicates where we're not able to detect methane production at all in the fluids. So this indicates kind of the opposite of our hypothesis that bicarbonate, even under hyperalkaline conditions, is still a preferred carbon source. And while formate and acetate can certainly supplement the methanogenic production of methane, it appears that truly bicarbonate is still the preferred source of food for these archaea. And so this gets us to the question of how we measure hydrogen uptake and methane efflux in an environment with abundant abiotic H2 and CH4 production. So this gets back to the slide earlier, where we're trying to, ultimately, we want to measure the biological transformation of these invisible metabolites, where we're not sure how much methane is coming from microbes, how much is coming from the formation, and similarly, we're not able to measure all the hydrogen that's lost. We're only able to measure the hydrogen that leaves the system on the surface. And so we thought that perhaps we could use our biomass turnover rates to get at this. And so we use this concept of microbial wattage to infer the fluxes of cryptic metabolites. And we're able to do this because of some foundational work by Tori Holer, who is a collaborator on this project, who showed that in his 2023 paper that the carbon turnover rates of cells shown on the y-axis and their mass-specific power, or the amount of energy that they are using to conduct their metabolism, are linearly related across many orders of magnitude. And what we're able to do is use this calibration to turn our biomass turnover rates that we measured empirically with the nanosims calculate mass-specific power, or the wattage of the cells. And if we know the power of the cells and the delta G of their metabolism, say autotrophic methanogenesis, we can then calculate the metabolite flux in terms of moles of H2 or methane per unit volume, per unit time, and kind of start to estimate these things at the landscape scale. And so what this data looks like is when you take mass-specific power requirements across Earth systems, this is just literature collected data. You can see that marine sediments have very low mass-specific power, and organisms in culture have very high mass-specific power. I put these up to bracket the data from our system that I will show you. You can see that we see a very large range of mass-specific power across the Oman system, where organisms at the fastest growing end are similar to microorganisms in soil or the very slowest growing cultures, but microorganisms at the slowest ends of growth are more akin to what you would see in rock-hosted subsurface and marine sediments that have been studied prior. And so once we take these mass-specific power estimates and we propagate them to the flux of metabolite that would be required to support those mass-specific powers, we see that both for hydrogen, which is shown here in the left panels, and the production of methane, shown here in the right panels, these fluxes can be substantial. And so we're looking at flux of hydrogen or methane in tons per cubic kilometer per year. And these numbers might not mean anything to you intuitively, but these fluxes are of the similar order of magnitude of what can be measured at the surface via gas monitoring of methane and hydrogen. It's also interesting to point out that these methane fluxes at the higher ends, so in the BA4A and BA1B wells, are similar if you consider them as emission factors. So like the amount of total efflux of methane from the subsurface, these methane effluxes would be of similar magnitude to brackish ponds or even cattle agriculture that you can see, that you can measure on the Earth's surface. And so it points to biological contributions to both hydrogen oxidation and methane production being non-trivial at the landscape scale. And so to summarize, our main conclusions are twofold. The serpentinite-hosted subsurface hosts microbial communities with extremely diverse rates of anabolic activity, but more importantly, extremely robust rates of anabolic activity. Oftentimes in these rock-hosted subsurface environments, we consider microbial life to be just holding on for dear life and doing what it can very slowly. But in this environment, we're able to see very fast rates of both anabolic and catabolic activity. And despite extreme limitation, dissolved inorganic carbon remains the preferred carbon source for both assimilation and methanogenic processes, which was surprising to us. And then our second key takeaway was that biological metabolite fluxes may be significant at the landscape scale. And we use these bioenergetic calculations to suggest that the serpentinite-hosted microbiome is contributing substantial methane efflux and H2 oxidation at the landscape scale. And so with that, I'll wrap up. I'd be happy to take questions, and I do want to acknowledge my former PhD advisor, Alexis Templeton, shown here in Oman, a former postdoc collaborator, Shrishti Kashyap, who worked heavily with me on this work. And yeah, I'll take any questions. We have a preprint out if you're interested in reading it.
Thank you so much for inviting me. It's great to share our research here. This work was done in collaboration with Chao Liang, my PhD classmate at Stanford, who is now a professor at Sichuan University in China. Also, John Nyken, who gave a talk here a couple of years ago, who is a researcher at the University of Oslo in Norway, and Wenbo Wu, who was here as an assistant professor, but just moved to Berkeley as an assistant professor, and Peter Kellerman, who many of you are familiar with, and finally, my advisor, senior scientist Rob Song, who is also here today. Okay, let's jump right into the topic. So I don't think anybody here needs me to emphasize the profound implications of serpentinization and the pyrrhotite carbonation. But I do want to emphasize the potential role that seismological techniques can play in this field, since most of the previous studies on pyrrhotite observation was done using chemical and biological methods. So the water rock reactions and the fluid transport associated with active serpentinization and carbonation happened in the subsurface, where it is very challenging to perform in situ measurements using chemical and biological methods, unless you happen to have a few boreholes, like the case of the MBO. But if there's a big if these processes generate detectable seismic signals or borehole acoustic signals, then it is possible to use the seismic data collected at the surface or borehole acoustic data collected in the boreholes to monitor these processes in real time. And this is the main goal of this main motivation of our study, that is to look for potential seismic and borehole acoustic signals generated by active serpentinization. And our study site is the NDO, which many of you are familiar with, and it's a multinational collaboration led by the co-PIs here. And the MBO established a few very closely spaced boreholes on a prototype terrain in the Sumaya ophiolite. And Rob, my advisor, conducted our seismic experiments in this area, where we have a few boreholes that are separated by just 100 meters. And this is the closed-in map showing our experiment sites. And the orange triangles are surface geophones, which measure surface velocity. In other words, it senses mainly seismic waves that propagate in the solid earth. And the two pink circles are the two boreholes, BA1A and BA1B, which Tristan just mentioned. And the two boreholes were 400 meters deep, with the water level at 15 meters deep. And in each borehole, we deployed 6 hydrophones spaced by 70 meters, with the shallowest hydrophone at 30 meters or 15 meters below the water table. And this is the first ever seismic and a borehole acoustic experiment dedicated to studying active serpentinization. And so all the signals that we see here were just, first ever. And in this data, we see an intriguing series of harmonic tremors. And we call it harmonic tremors because its power spectral density, which is basically a Fourier transform of a segment of the time series, consists of a series of very narrow spectral peaks whose frequencies are integer multiples of fundamental frequency. And in this case, we have observed in total 12 modes, ranging from the lowest one at about 25.5 hertz to 190 hertz. And all of them are very regular space with the frequency spacing of about 12.75 hertz. But an interesting fact is that there are a couple of modes that are not observed, which are marked in question marks here. And most importantly, somehow the fundamental mode was not observed. And that's kind of unusual because in the resonance of many so in a resonance model, in most of the cases, the fundamental mode is the most prominent, but here it's not observed. So this is just a 1 or 5 minute geophone signal. And if we extend that to the 9 months hydrophone data, sorry, I forgot to mention that the hydrophones were deployed in the boreholes for 9 months. And for the last 21 days of the hydrophone deployment, we further deployed the geophones on the surface. So the geophones were there for 21 days, whereas the hydrophones were there for 9 months. So on the top, I'm showing the spectrogram of the record of the 9 months record of one hydrophone in borehole BA1A. For those of you who are not familiar with this representation of the data, we are just dividing the 9 months long time series into 5 minute long time windows. and do Fourier transform of every one of them. We then just put them one by one, side by side, to make this image where the color indicates the spectral power. And in this plot, we clearly see this nicely gliding spectral line that was active for the entire 9 months. And we can clearly see that it shows a diurnal cycle, which is more clearly seen in this zoom in window of the 21-day period. And this is one of the hydrophones. On the left-hand side, I'm showing the data for the same 21-day period recorded by two surface geophones, which showed exactly the same frequency variation. And this suggests that this signal is indeed a true signal, not an instrument noise, which sometimes do exist on seismic or acoustic data. And remember, we have 12 observer modes, so we can extract the frequency of each mode for the 21-day period of the geophone observation and plot it in them as a function of time. And here I'm showing all of them with the color, meaning the average frequency of each mode. The vertical white and the gray bars mark days and night, so that you can more clearly see the diurnal frequency variation. And here you clearly see that all the modes show a completely synchronized frequency variation. And for the lower modes, especially mode 2 and mode 3, they are observed for the entire 21-day period. But for the higher modes, they are only observed intermittently. And for the highest modes, they are mostly observed at night, which may be due to higher noise level at the site, because the wind is stronger in the day and lower in the night. And the next question is, where is this tremor source located? So this is very challenging to work out, because unlike regular earthquakes, which have clear onsites, these tremors are continuous. So there is no way to pick an arrival time whatsoever. However, fortunately, we have a very dense surface observation network. So we can use the differential phases between pairs of stations to derive a propagation vector for each station trias, shown in gray here. And the arrows show the local wave propagation vector for each station triad. The direction of the arrow points to in which direction the wave propagates, and the color indicates the apparent velocity of the wave field. And we see that there is a significant scatter in all the arrows, but in general, the arrows point northward for both the subarray A in the south and subarray B in the north. And moreover, we see a significantly higher propagation velocity in array B than array A overall, and this is consistent with the subsurface condition of the two sites. The array B is located array A is located on a soft segment, which is relatively uniform. So that facilitates the propagation of slow surface wave. Whereas for array B, some of the stations were located on bad rock, which facilitates the propagation of fast body waves. So this tells us that the source likely located south of our site, but it doesn't really tell us the distance. However, since we have observed frequencies of modes with a frequency higher than 150 hertz, that suggests, and those waves attenuate really fast with distance. And this tells us that the source must be just a short distance away from our south. So combining these two pieces of evidence, we conclude that the source is located a short distance south of the site. The next question is, arguably the most important one, is what is the source mechanism? To answer this question, let's first look at some of the similar signals observed by previous studies. So these kind of harmonic tremors were commonly observed in systems hosting active subsurface fluid flow, such as active volcanoes. Here is an example from the Lascar volcano in Chile, and the x-axis, and this is a spectrogram like the one I showed you previously, although it kind of rotated by 90 degrees, where the y-axis is time and x-axis is frequency. And on this, you can clearly see these gliding spectral lines that are completely synchronized in their frequency variation. And it lasted the almost entire 2 hours of this data segment. And in addition to that, similar signals were also observed in a hydrocarbon reservoir undergoing hydraulic fracturing treatment. This is the data presented by this 2014 paper, where you also see these gliding spectral lines, which the author found that correlate with the various agents injected into the reservoir, which were plotted on top of each panel. So these results suggest that our harmonic tremors are probably also caused by active subsurface fluid flow. And considering that our site is in an active serpentinizing terrain, the fluid that is moving underground is likely free gas. And we already know that there is free gas migrating at our study site. So this is the work done by my colleague, John Nyken, in 2022. I think he probably talked about this a few years ago on this seminar, where he showed that the borehole hydrophones detected swarms of gas bubbles being injected into the boreholes during the 9 months period. So on this plot, the top one shows the waveforms of one bubble injection, which clearly initiated on the top of the borehole and propagated downward and reflected back. And the bottom panel, it shows how the waveforms of the bubble swarm look like in a 12-hour time period. And on the right-hand side is what we see is happening, which is there is a trap and release process at a point on the borehole where gas is accumulates and released periodically. And this causes the episodic injection of the gas bubbles. Therefore, considering this, we think that the tremors we observe may be generated by sustained free gas flow through a fracture at 15 to 150 meter depths. So how do we come up with these numbers? So 15 is the water level, is the water table depth, and 150 is where previous works identify a steep drop in permeability in the boreholes. And this is why we think that the source is probably located in this depth range. And assuming this model, we can infer the physical properties of this fracture using the observed quality factors and frequencies of our harmonic tremors. So on the top, I'm showing the relation between quality factor and gas volume fraction. So in this model, the quality factor, which measures how fast the energy dissipates from this resonator, increases with increasing gas volume fraction. And the green and the purple shows two models made for sources located at 15 meter depth and 150 meter depths. And the orange bar shows our observed quality factors, which are very high. They are on the order of a few thousand. And by comparing these two, we can see that the gas volume fraction in this fracture must be above 70%. And then we use the frequency of our observed tremor to infer the dimension of this fracture. So the frequency of the tremor depends on the dimension of the fracture and the impedance contrast across the fracture wall, which in turn depends on the gas volume fraction. And since we have already determined the gas volume fraction to be above 70%, we can see that we are in this range of the plot, which suggests the fracture dimension of a few meters to a few tens of meters. Okay, so this is all about our tremors, and if we combine it with the bubble injections that John observed before, we can have this conceptual model of how free gas migrates in the subsurface of our site. On one hand, you have episodic free gas releases into one of the boreholes. And on the other hand, you have continuous gas flowing through an underground fracture, which generates our sustained harmonic tremors. These inferred processes suggest that the aquifer of the study site is probably very rich in free gas. However, that contradicts apparently with previous geochemical analysis, which found the borehole fluids in the two boreholes to be undersaturated in both hydrogen and the methane. And so if we assume that both the geochemical and our geophysical results are correct, then we have to find a way to reconcile them. And the only way we can think of that can reconcile the two results is that the gas and liquid phases in the aquifer may be out of equilibrium, which means that free gas may flow along a certain well-established path, whereas and outside those major free gas pathways, the aquifer may be undersaturated in methane and hydrogen. And if our model is correct, then seismological techniques may hold great potential to identify those elusive gas migration pathways, as shown by my work and John's work. And this is all I have today, and happy to take questions.